Notice the detailed taxonomy here. As I enlarge this section, you can see how the frontal lobe connects.
Right, that clarifies the pathway.
The human central nervous system is organized into two distinct yet structurally and functionally interdependent compartments, the gray matter and the white matter. The cerebral white matter serves as a vast, highly anisotropic network of myelinated nerve fibers. Furthermore, the white matter microenvironment is sensitive to acute physiological stress. For example, within 1 week of a traumatic event, the myelinated axons within the cerebral white matter are organized into highly structured pathways, or tracts, which are classified into 3 distinct categories. Projection tracts interconnect the cerebral cortex with subcortical nuclei, basal ganglia, brainstem, and spinal cord. We will review the corticospinal, The anterior limb primarily carries the anterior thalamic radiation and frontopontine tracts. The genu contains the corticobulbar pathway. The internal capsule is supplied by the small, perforating lenticulostriate and thalamostriate branches of the middle and anterior cerebral artery. The arcuate fasciculus connects Wernicke's area in the temporal lobe with Broca's area in the frontal lobe, critical for language processing. Another association pathway is the cingulum, which travels within the cingulate gyrus to connect the cingulate cortex. Additional pathways include the uncinate fasciculus and the inferior longitudinal fasciculus, connecting the temporal and occipital lobes. Structurally, the corpus callosum is divided into 5 regions: rostrum, genu, and body. The rostrum is the thin, anterior-inferior segment. Isthmus, the narrow segment located where the fornix meets the undersurface of the corpus callosum. It separates the anterior associative segment. The corpus callosum serves as a major pathway for interhemispheric communication. In clinical oncology, malignant brain tumors, myelin sheath synthesis in the central nervous system is a tightly regulated, multi-step process. Oligodendrocytes develop from a lineage of proliferating. This compaction process requires specific myelin proteins, which are synthesized locally at the axon-glial contact site via the transport of mRNA transcripts. Proteolipid protein, or PLP, is the most abundant protein in CNS myelin, playing a key role in the compaction of the extracellular leaflets. CNP and MAG are crucial for the metabolic and structural support of axons. Genetic depletion of CNP or PLP does not prevent remembrance. The axonlemal domain includes scaffolding proteins like ankyrin B. At the paranode, key cell adhesion molecules The node is stabilized by a specialized extracellular matrix, rich in chondroitin sulfate proteoglycans. During clinical remission, however, resident microglia localize to the nodes, clearing damaged myelin debris and releasing signals to promote repair. In MS, autoreactive CD4 plus T helper cells and CD8 T cells cross a compromised blood-brain barrier. The loss of metabolic support from the myelin sheath impairs mitochondrial function within the axon, leading to chronic energy deprivation and calcium overload. Leukorrhosis represents a chronic, ischemic degenerative process driven by cerebral small vessel disease in the aging brain. The medullary arteries are end arteries. They run perpendicularly through the cortical layers to reach the paraventricular region without forming anastomoses.
Duration. Females show higher extracranial arterial tortuosity, a greater volume of white matter hyperintensities, and elevated susceptibility.
Diffuse axonal injury, rotational shear, and proteolytic collapse.
Primary axotomy. The mechanical strain stretches the axonal membrane, causing microscopic breaks in the stiffest structural elements of the cytoskeleton, the microtubules.
Stage Y. Histological evidence of axonal injury is confined to the lobar white matter of the cerebral hemispheres.
Stage 3. Involvement of the lobar white matter and corpus callosum, with additional focal lesions in the dorsolateral rostral brainstem trite. In contrast, immunostaining for beta-amyloid precursor protein is highly sensitive, revealing axonal transport disruption within 2 to 3 hours.
Standard computed tomography is insensitive to DAI. MRI is the modality of choice. SWI reveals traumatic microbleeds. Ultra-high field 7 Tesla SWI increases TMB detection.
Wallerian degeneration is a stereotyped, anterograde degenerative process that occurs in the distal segment of an axon following physical transection.
All right, sir, gently scoot towards the machine. We've got you. Take your time.
Because of this high specialization, the white matter is vulnerable to diverse pathological insults. In multiple sclerosis, autoimmune attacks disrupt the myelin sheath. Understanding the structural organization, cellular dynamics, and radiological correlates of these conditions is critical for developing targeted therapies.